Hello everyone and welcome to another MCAT ACE lecture. In today's video we are going to discuss the reproductive system and the difference between mitosis and meiosis. So I've divided this lecture into two parts. In part one we are going to review all the topics that are shown here in the red box, which includes the difference between diploid and haploid cells, the mechanisms of mitosis and meiosis, how eggs and sperms are being produced, the other terms for which is oogenesis or spermatogenesis, and then go over the functions of different reproductive organs, and in the end discuss how male and female sexual organs develop from their embryonic structures. Alright, and then in the second part we will go over the male and female reproductive structures and the hormonal controls of the male and female systems. And then finally we will discuss integration of our reproductive system with the nervous control, how basically the reproductive hormones affect our brain. And then from there on, we will, as always, conclude our video with some practice questions. Alright, so here starting with the first part of this video, let's go over the difference between diploid and haploid cells. So basically, cells that have two pairs of each chromosome, the other name for which is homologous chromosomes. These are called diploids. And here, like for instance, you can see a diploid zygote right here. It has a pair of red chromosomes, a pair of orange chromosomes, and a pair of green chromosomes. So these two red ones, for instance, are homologous chromosomes. And so since there is two pairs of each chromosome, therefore these are called diploid cells or 2N. And these diploid cells can replicate by mitosis in order to yield more diploid cells, which are also 2N. On the other hand, we have some germ cells, like for instance, the cells in the testes or ovaries that are capable of producing gametes. In males, it's called sperm, and in females, it's called an egg. And these gametes are haploid, meaning that they only have one copy of each chromosome. So here you can see that we only have one orange, one green, and one red. Therefore, once these two gametes combine, a sperm is combining with an egg, that's the time that we get a zygote, which is a diploid. And this zygote will undergo multiple replications until eventually it gives rise to the organisms, like humans, for instance. And so our body has 2N chromosomes, except for the haploid sperms or haploid eggs that are produced by germline cells. So humans have 46 chromosomes. Or the other way to say it is that they have 23 pairs of homologous chromosomes. Sperm and egg, on the other hand, in humans, each of them contain only 23 chromosomes. And so therefore, since they are haploid, it's called N. Now let's move on to the cell cycle and discuss how cell can undergo division. So the cell cycle is divided into all the phases that are shown right here. So this is basically a summary of different phases of the cell cycle. So the first phase is G0, or the quiescent state. This is the state at which time there is a cell cycle arrest. There is no plan for division. And so the other name for it besides G0 or quiescent state is non-growing phase. And during this time, cells will produce proteins for various different functions. Right? Like for instance, think of your muscle cells or your nerve cells. They do not normally divide, so they are in their quiescent state. But they still do their function. They still produce the protein they need to do, right? So they are in the quiescent state. They are not growing, but are producing proteins needed to carry out their functions. The next phase is interphase, which is shown right here in the light blue color. And interphase is actually the time during which cell grows and is preparing for the next step, which is mitosis, and that's division of the chromosomes, and then finally cytokinesis, which is division of the cytoplasm. So interphase is actually the growth phase, and interphase is divided into three phases itself. G1, during which time cell grows, in size, and it produces more ribosomes and proteins in preparation for cell division. Then we have the S phase, which is the phase that DNA replicates. So here during the S phase we have DNA replication, and then finally we have G2 phase, during which time the cell continues to grow. So here cell continues to grow, and then organelles will also divide. Next we have mitosis, during which time the chromosomes will separate from each other, and mitosis is divided in 4 different phases, so let's review those. But before I go over those, I'm going to provide you with a memory aid so that you remember the order of these phases. The way I memorize it is called PMAT. So you know how we are taking MCAT, the order of the phases in mitosis is called PMAT. Prophase, metaphase, anaphase, and telophase. So now let's go over each phase and discuss what happens during every one of them. So the very first one is prophase, that I've shown right here, the image for you. During this phase, chromatids pair and attach to one another by centromere. So there we go. So chromosomes attach to each other by centromere, and they become visible and condense into chromosomes. Alright, so chromatids pair with each other by centromere, they become visible, and they turn into chromosome. At the same time, nucleus starts to disintegrate. So here you can see that I've shown the nucleus membrane with a dashed line. It's because nucleus start to disintegrate. And then one other change that you will see here is that centrosomes will start to move to the opposite sides and form spindle fibers. Alright, so other changes is nucleus disintegrates and spindle fibers are formed, and finally centrioles move to opposite poles. The next phase that we are having is called metaphase, and metaphase is actually the time where the paired chromosomes come to the center of the cell, which is called metaphase plate. So during this time, pairs of chromatid line up at the center of the cell. So chromatids or chromatid pairs line up at the center of cell, which is called metaphase plate. And during this time, centromeres of each chromatin becomes attached to 2 spindle fibers, right? So here you can have each one of them is attached to 2 spindle fibers, and these spindle fibers attach to the kinetochore of the centrosome. So here we have the chromosome. At the center we have centromere, and spindle fibers will attach to the kinetochores. So here we have the spindle fibers attaching to the kinetochore of every chromosome. And then during the next phase, which is anaphase, sister chromatids are starting to pull apart from each other. So as these spindle fibers start to shorten, sister chromatids will start to move to the opposite pole of the cell. So here we have in anaphase, sister chromatids will separate into opposite poles of the cell. Alright, and then at the same time the centromeres will start to divide. The final phase is telophase, during which time chromosomes now arrive at the opposite pole of the cell. So here you can see that chromosomes have arrived at the opposite pole of the cell, and this is now the telophase stage. So at this time they will start to uncoil, spindle fibers will disappear, and a new nucleus membrane will be formed. So here you can see that now we have a new dashed line around it. It's because a new nucleus membrane is forming. So again, telophase, what happens here? The chromosomes have arrived at opposite poles. So chromosomes at opposite poles, they start to uncoil, spindles disappear, and a new nucleus is being formed. Now the next stage is cytokinesis, which I've shown right here for you. So after the chromosomes are now divided, the next stage is cytokinesis, which is shown right here in this dark gray shade. And cytokinesis is division of the cytoplasm. Because think of it, we've already divided the chromosomes, but we still need to divide the cytoplasm. So cytokinesis is the stage where cytoplasm divides into two separate cells. All right. In animals, what happens is that a cleavage furrow will be formed at the center of the cell in order to help with the cell membrane to pinch into the cytoplasm. So here you can see that we have a cleavage furrow in animal cells that happens during the cytokinesis stage. In plants, on the other hand, a new cell wall is being assembled at the center of the cell. All right, so here I have again the summary of all the stages that I described for you. The cell cycle, G0, then we enter the interphase, G1, S, G2, and then finally chromosome division during mitosis, and then cytoplasm division during cytokinesis. And then here is also a summary of the phases of mitosis, which was PMAT. Now, the next concept is meiosis. And basically, meiosis is what germlines are going through in order to produce sperms and eggs. So you know how we discussed 2N cells undergo mitosis in order to yield 2N cells. Now, germline cells, which are 2N cells, they undergo meiosis in order to yield haploid or N cells, example of which includes like sperm and eggs. All right, so here we've already discussed the mitosis phase. I'm not going to go over this phase. I just showed it on the right side so you can compare it to the phases of meiosis. So meiosis actually happens in two separate steps. One, we call it meiosis 1, which has its own prophase, metaphase, anaphase, and telophase stage. And then in meiosis 2, again, cells are going through another stages of prophase, metaphase, anaphase, and telophase. So if you think of it, DNA during meiosis has been replicated only once, but then we are having two divisions. Okay, here we are having first division, and then in meiosis 2 we have the second division. So what happens with mitosis? We had one replication and one division, as a consequence of which 2N yielded 2N. In meiosis, we have one replication but two divisions. That's why 2N will yield 1N, because we are having one extra division. All right, so now let's go over all the phases of meiosis and discuss what is recombination that is happening during this phase. Alright, so meiosis starts with interphase, during which time DNA, centromeres, and centrioles will duplicate. Then we have prophase 1. So similar to mitosis, during this time nucleus disintegrates, chromatids become evident, and centrioles move to the opposite side of the cell and form spindle fibers. At this time now, homologous chromosomes form a tetrad and undergo recombination, the other name for which is crossover. So inside prophase 1 right here, cells will form a tetrad and undergo recombination. And here in the next step, I've described the recombination for you, so let's review that right now. So here during prophase, a tetrad will be formed, okay? And by formation of this tetrad, homologous chromosomes can undergo recombination, or the other name for which is crossover. Okay, so once again, when does recombination or crossover happens? It happens during the prophase 1, or in other words, in the prophase stage of meiosis 1. And now, what's the purpose of recombination? Basically, recombination allows for genetic variation in offspring, because what it does is that it combines chromosomes with each other and exchanges. Imagine that you're having a deck of cards and you shuffle them together. Now you're having more variation. So that's the purpose of having recombination, so that the offsprings, they won't be all identical. They would have some variation. And the term chiasma referred to the site where the chromosomes exchange their genetic material during recombination. So here you can see that we have one blue chromosome here. The two sister chromatids are attached to each other by centromere. And then we have another homologous chromosome right here that they have the two sister chromatids again attached to each other by a centromere. During recombination, they form a tetrad, and then at chiasma is the site where they exchange their genetic material. So you can see that part of the chromosome from the blue part has been transferred to the pink chromosome, while part of the pink chromosome is transferred to the blue one. So here we had genetic exchange, which gives us more variation in offsprings. Okay, now let's go back to our table here. So during prophase 1, that's when a tetrad will be formed and cells will undergo recombination. Next phase is the metaphase 1. You can see that the exchange has already happened. I know that the quality is a little low, but I can zoom in right here for you. So here you can see that I've already shown you that the recombination has happened. Now during metaphase 1, similar to what had happened in mitosis, the chromosomes will line at the equator and spindle fibers will attach to their centromeres. Next we have anaphase 1. Again, similar to what happened in mitosis, the homologous chromosomes are pulled apart to the opposite poles. So here the homologous chromosomes are being pulled to the opposite sides. And then finally we have the telophase, during which time, again similar, everything is very similar to what happened in mitosis. During telophase, chromosomes will uncoil, they become evident, spindles will disappear, and a new nuclei will be formed, which I've shown here in dashed color for you. And then the cell will also divide via the cytokinesis. So at this stage, now the homologous chromosomes have been divided and moved to the opposite side. So you know how with cells, we mentioned that cells that have two homologous chromosomes are called 2N. This cell, the homologous chromosomes are separated, so we already have 1N. But each chromosome still has two sister chromatids. So this cell is 1N, 2C, because it has only one homologous chromosomes and two sister chromatids. All right, next we have repeat of the cycle. Again, we have prophase 1, during which time nucleus disintegrates, chromatids become evident, and centrioles move to the opposite poles. And then following that, we have metaphase 2, chromosomes line up at the center of the cell, spindle fibers attached to them. Then we have anaphase, where chromatids are being pulled to opposite poles. So here note that sister chromatids are now being separated from each other, as a consequence of which now the four cells that we got here are 1N, 1C, because each cell has only one chromatid of every homologous chromosome. Here we only have 1, 1, 1, 1. So 1N, 1C we are having. Okay, and so as a consequence of meiosis, one cell has yielded 4 haploid cells, versus in mitosis, one cell yields 2 diploid cells. Okay, and so that's about it regarding discussion of the difference between mitosis and meiosis. Now let's go over a little more detail discussing how an egg is being produced. So oogonium is basically the cells that give rise to an ovum or an egg, and it's a 2N, 2C cell, meaning that it has diploid or homologous chromosomes, and there is only one sister chromatid in each of these chromosomes. Following replication and during the interphase stage, the chromatids will duplicate. So now every chromosome has sister chromatids They are still diploid because we only have one pair of homologous chromosomes, but their chromatids have doubled. So now we have 4C. During meiosis 1, homologous chromosomes will separate from each other and yield 1N, 2C. And during the second meiosis, sister chromatids will separate from each other and yield an ovum that is 1N and 1C. So the very first cell was called primary oocyte, that was 2 and 4C, and then after meiosis 1 it was called secondary oocyte, and then finally after meiosis 2 we are having ovum. Now if you've noticed, in females one cell gives rise to only one ovum, and it's because some of these cells will degenerate and form polar bodies. And polar bodies are being formed due to the asymmetric cell division, because once an egg is being formed, the body wants to get as much cytoplasm and organelles for that egg, because that egg is going to combine with the sperm. Sperm doesn't have really much material. Indeed, all our mitochondria comes from mother's egg, right? That's why we say mitochondrial disorders are being transmitted only from mother. The father doesn't contribute to mitochondrial disorder. And the reason for that is all the mitochondria from combination of an egg and this very tiny sperm, all the mitochondria is inside this egg. So that's why mitochondrial disorders are being transmitted from mother. Now, in order to get all the mitochondria and all the organelles, the cell doesn't want to go through like symmetric division. Not much would be left here. So here, during the formation of an egg, cells will undergo asymmetric division, where one cell will have majority of the cytoplasm. The other one, which is called polar body, will have a small amount of cytoplasm and will undergo degeneration. So therefore, with oogenesis, one cell will give rise to 3 polar bodies that will degenerate and 1 egg that can now combine with the sperm and form a zygote. Next, we have the spermatogenesis, which is formation of sperm from spermatogonial stem cells. So this is very similar to what I've just discussed with you regarding formation of an ovum or an egg, except that there is no polar body formation. So during spermatogenesis, one stem cell yields 4 different spermatids. And again, it's exactly a similar process. So let me mention that this process takes place in the seminiferous tubules. So that's where sperms are being formed. And so in the very first step, there would be replication. So here we are having a stem cells that is diploid, and that's why it's called 2N, because we are now having homologous chromosomes. Every chromosome is in pair. And then the chromosomes only have 1 chromatid. So you can see each chromosome has only 1 chromatid. Now, after the first division, that cells undergo division and their interphase and replication of the DNA, they're still having 1 pair of every chromosome. So they're called homologous chromosomes, but the chromatids have duplicated. So now we are having sister chromatids. So here you can see 1 and 2, these are sister chromatids. 1 and 2, these are sister chromatids. Now in this other homologous pair, we have 1 and 2, these are sister chromatids, and then another sister chromatids in the other chromosome. So this is called a primary spermatocytes. After meiosis 1, homologous chromosomes will separate from each other. And so now we are having 1N, 2C, and this is called a secondary spermatocyte. And then finally, during meiosis 2, the sister chromatids now separate from each other, yielding 4 spermatids, which are 1N, 1C. All right, so again, just pay attention that the diploid definition refers to the presence of homologous chromosomes. So spermatogonium and primary spermatocytes are diploid, while secondary spermatocyte and spermatid are haploid. Now, what happens after these spermatids are formed? These cells are still immature, and so now they need to develop into a mature spermatozoa, and this process is called spermiogenesis. Genesis, meaning that these spermatids will now develop into a mature spermatozoa. spermatozoa. Okay? And then during spermiogenesis, the changes that you will see is that the spermatozoa become elongated. It will start to develop a flagellum, a midpiece, and then finally its head will start to develop an acrosome. So now let's go through details about the function of every one of these structures. Alright, so first let's talk about acrosome. This is basically an acidic secretory vesicle, and it's derived from Golgi apparatus that contains the hydrolytic enzymes required for the sperm to penetrate the wall of the egg. So this acrosome will help the sperm to penetrate into the wall of the egg and enter the egg. The middle part, which is called midpiece, is containing axonemes that are surrounded by mitochondria. And so this structure is generating enough energy in order to promote the flagellar motility. And then next we have the flagellum, which contains axonemes that are surrounded by a fibrous sheath, and the function of the flagellum is to propel the spermatozoa by its oscillating waves. So you can see how it's having the oscillating waves. So by having this oscillating waves, it will push the sperm forward. Now here I would like to mention a quick point, and that is prokaryotes like bacteria can also have flagellum, but the flagella of the eukaryotic cell is more advanced, because in eukaryotes it has a bending movements and it's kind of like a Whipple motion. In bacteria it can move either in clockwise or counterclockwise rotations. So the type of movement that is seen in the flagellum of the bacteria is rotatory fashion, while the one that we are seeing with the sperm is running by bending movements. So here next I have a summary of everything that we just discussed regarding the sperm formation and the spermiogenesis. So here we just discussed that in the seminiferous tubules of the testes. So here we have a testes that is made of a lobules, and we have shown you a cross section of one of these seminiferous tubules, and it's inside the seminiferous tubules that spermatogonia, which is our stem cell, will undergo meiosis 1 and meiosis 2 in order to yield spermatids. And then spermatids will undergo spermiogenesis in order to produce a mature spermatozoa or sperm, which is now capable of fertilizing an egg. And we discussed the functions of the acrosome, which contains the hydrolytic enzymes for penetrating the egg. It has the nucleus right here, which contains the genetic material. So here is where our genetic material is. Then we have the midpiece that contains mitochondria as well as the axonemes in order to generate enough energy for moving the flagellum. And then finally we have the flagellum that moves with bending motions. Alright, now let's move on to the next section, which is to discuss the functions of different reproductive organs. So first I'm going to give a quick summary of different reproductive organs, and then in the next lecture I will go in a little more detail regarding every one of these structures in males versus females. So the first reproductive organ is gonads, where in females it's called ovaries. So here we have ovary in female, and in males it's called testes. And their function is to produce sex hormones and gametes. So ovaries and testes will produce sex hormones and gametes. Like for instance, here they produce sperm, and in female it produces an egg. And the amount of sperm that is produced by testicles is about 1 billion gametes per day. So 1 billion. Versus in females, ovaries release 1 gamete per menstrual cycle. So there will be 1 egg per menstrual cycle. So in female, oocyte travels all the way from fallopian tube into the uterus, and it's inside the uterus that implantation happens, and the fetus will get its nutrition from mother. In males, on the other hand, it's a little longer path. So we discussed how sperms were initially formed in the seminiferous tubules. From here on, they will be stored in the epididymis until they are released. So once they are released, they will go through vas deferens, and from here on, they would go to the ejaculatory duct, and then finally urethra and the penis. So the memory aid that I have here for you is called 7-up, in order to remember the order of events that happens here. From where does the sperm move? So the S stands for seminiferous tubules, which is the structure inside which the sperms were formed. Then we have epididymis, which is the structure inside which the sperm is being stored. Then we have V standing for vas deferens, E stands for ejaculatory duct, N is nothing, so you don't need to place anything for here. N is nothing, we just put it here for ease of the memory aid. Then we have up, so U stands for urethra and P stands for penis. All right, the next structure in the reproductive organs is the accessory glands and organs. So in males, it's the prostate gland and seminal vesicles that secrete fluids that mix with the sperm during ejaculation. And then there is also bulbourethral gland that will produce an alkaline lubricant in order to help the sperm to survive the acidic environment of vagina. So in other words, the accessory glands in males include prostate, seminal vesicles, and bulbourethral gland. And we will go in a little more detail regarding every one of these structures in our next video. Here I'm just summarizing all the findings. In females, on the other hand, uterus is the structure that provides support and protection for the growing embryo. And then finally, we have the perineal structures or external genitalia. So now let's go over the similarities and differences between the male and female reproductive systems. So both male and female reproductive systems develop from similar embryonic structures. So they all have similar embryonic origin. And so therefore male and female sexual organs have counterparts, and we will discuss that at the end of this video. Both systems also contain gonads. So both have gonads, which is testes in males and ovaries in females. And they both also produce sex hormones. Like for instance, androgen and testosterone in males versus estrogen and progesterone in females. And then it's secretion of these sex hormones that induces maturation of male and female reproductive organs. So sex hormones in both males and females led to puberty and sexual development. Again, both male and female will undergo meiosis. So that's another common feature that we see in both of the reproductive system. And then both systems rely on follicular stimulating hormone and luteinizing hormone in order for regulate secretion of different hormones and different products. Now let's talk about the differences between these structures. So one main difference is that testes can produce sperms without limit. So we just discussed as much as 1 billion sperm can be produced per day, versus ovaries are limited. They can only produce 1 egg per menstrual cycle. And this is because of the fact that the number of eggs that are produced are fixed before birth. So there is a limited number of eggs. And so from menarche to menopause, since we have a limited number of eggs inside each ovaries, there is only 1 oocyte that is being ovulated per menstrual cycle. So in other words, the body knows that it's limited, has to be frugal, cannot release like 1 billion like the testes does. So there is a limited supply of eggs. And then the other difference is that, as we just discussed, meiosis in males, every meiosis produces 4 sperms, versus in females, it only produces 1 egg, which is functional, and then 3 polar bodies, which are small and non-functional. All right, so that summarizes the similarities and differences between the male and female reproductive organs. Now let's move on to the next section and discuss how the embryo is having similar structures. The same structure is in both male and female. How does one fetus with the Y chromosome determines to develop male organs versus the other one that doesn't have Y chromosome will develop female organs? So the embryo is bipotential, meaning that it can develop into male and female depending on the factors that are available. By default, the embryo is destined to become a female. However, if there is an SRY gene present, which is found in the Y chromosome of males, that's the time that the path of the organ development switches from becoming female into becoming a male. And so what happens? SRY gene, it contains a testes determining factor, which induces differentiation of primitive gonads into testes. So instead of forming ovaries, now testes are formed in the embryo. And now these testes will produce testosterone via the Leydig cells and Mullerian inhibiting factor via Sertoli cells. So what the Mullerian inhibiting factor does is that it will cause degeneration of paramesonephric, or the other name for which is Mullerian ducts. And so by doing this, female organs will not develop. So normally we need development of paramesonephric or Mullerian duct for female structures to develop. But since we had testes, it has secreted the Mullerian inhibiting factor that inhibited development of female reproductive organs. On the other hand, Leydig cells has produced testosterone, which enhances development of mesonephric or Wolffian. The other name for it is Wolffian ducts into male internal genitalia. So now we are having male reproductive organs that are developing. And then at the same time, testosterone will be converted into dihydrotestosterone, or DHT, with the help of 5-alpha reductase. And this DHT induces the formation of the male external genitalia as well as prostate gland. All right, so this whole thing happened under the circumstances where the SRY gene was present on the chromosome. So now what would have happened if we didn't have this SRY gene? So if we didn't have the SRY gene, then what would have happened was that the primitive gonads would have developed into ovaries. And then ovaries would induce the Mullerian or the other name for which is paramesonephric ducts to develop into fallopian tubes, uterus, and parts of the vagina. And then since there was no testosterone, mesonephric or Wolffian ducts would have degenerated and no male organ would have been formed. All right. Now moving on to the next section, which is the male and female counterparts. So as we just discussed, both male and female have developed from similar structures. So let's discuss how different embryonic structures gives rise to male and female reproductive organs. So first we have the genital tubercle, which is shown right here in red, and it gives rise to glands of the penis in males or the clitoris in females. All right, so male glands of the penis and clitoris in females, these are sexual counterparts. Then we have the urethral folds and groove, which is shown right here, the urethral folds and groove that gives rise to the shaft of the penis or labia minora and vestibule in females. And then finally we have the labioscrotal swelling. So here is the labioscrotal swelling shown right here. That gives rise to the scrotum in males or labia majora in females. And so that concludes the first section of our video. Now I'm going to pause the video and move on with the second part, which we will discuss the rest of the topics that are shown at the bottom here. All right, so thank you all for listening and see you for the next video.